Hi everyone! In this video, I'll be showing you how to use Kolmogorov Complexity to assess how complex a DNA sequence is. So we'll be reproducing a research paper that I found, and the whole code is available on my GitHub account. So one of you asked me help with this particular topic of K complexity and DNA sequences, but this isn't the first time that someone asked me about K complexity. So I've decided to make like a thorough video just to show you the whole process, and this doesn't have to be DNA sequences. Basically, any type of sequences will do. So if you prefer to go through a blog post instead of a video, I have this on the description of the video. So the question I received is the following. So please, I want to use Kolmogorov Complexity to compress a DNA sequence using Python. Can I have someone that can guide me with the code? So the first part is I want to use Kolmogorov Complexity. So for those that don't know, Kolmogorov Complexity is a sort of complexity that is kind of used in many fields. However, the important part here is that this metric is actually theoretical, so you can just use it. So the middle part of the question says Kolmogorov Complexity to compress. So this is a bit weird because the measure is theoretical, but yeah, usually this is where the confusion arises. So we're gonna kind of untangle what is the relation between the compression and the complexity, and also how to practically use a measure that is purely theoretical. So we'll see in a few that the actual implementation that you can use for Kolmogorov is Lempel-Ziv, and this will tie everything together. So finally, the last part of the question is compress a DNA sequence using Python. This last bit hint toward using Python and DNA sequence data structure in order to get the complexity metric out of all of this thing. So it's important here that we understand the data format and how to play and wrangle with the data, because you see that there's like multiple different parts to get this right. I'll provide some working code to reproduce roughly some paper that I found that is actually doing the same kind of workflow. To make sure that you understand well what's going on, at the end we'll go through a 6-step. Step 1, first we'll go through what a DNA sequences look like for real, and how to load that into a Python analysis. So step 2, we're going to go through the explanation of exactly what Kolmogorov complexity is. So step 3, we're going to walk through a paper that highlights how Kolmogorov complexity is used with DNA sequence, and this will be the basis for the code afterward. So step 4, we're going to go through an explanation of what Lempel-Ziv complexity is and what is the relationship with Kolmogorov complexity. So in the step 5 of this video, we're going to go through an overview of the data pipeline from the raw DNA sequence to the complexity metric output. And finally, for step 6, we're going to go through the full walkthrough of the code with Python and yeah. So let's jump right into the DNA. All right, so step 1. So what is a DNA sequence and how does it look like? So DNA molecules are both incredibly complex and beautifully simple. However, usually when we're talking about using Kolmogorov complexity to analyze them, we restrict ourselves to the nucleotide sequence that is adenine, thymine, guanine, and cytosine. So all of the added complexity of methylation and epigenetic and all that jazz, we're going to just assume that it doesn't exist. So basically we have a sequence at the data level that is A, T, C, and G, and that's it. So it's a bunch of that. Yeah, that's all there is. It's just a big long string sequence of these 4 letters. The arrangement of these characters and their pattern is what encode the protein that makes cells so complex. So going on Kaggle, it's pretty easy to find some DNA dataset which we can start to play around with. However, we're only trying to calculate complexity here, so we don't need to do too much background information about where the data came from, because we're just trying to get like a bunch of characters that is A, T, C, and G in all of the different permutations, and then from these blobs of character determine a measure of complexity. So the basic assumption about DNA is that the sequence of character of a given DNA snippet is closely related to how complex it is. So what I mean by that is, if we had the full DNA snippet that contained only one character, let's say A, all the way through, it's safe to assume that this snippet is not complex and doesn't hold much information. However, a more complex DNA sequence with like a random mix of all of these different patterns of A, T, C, and G is assumed to be more complex, so to hold more information. So this is where a complexity measure is important, which can quantify this difference. So here you see I'm making like the relationship between complexity, information. It's more complicated than that, but let's assume that these two are related. So there exists a few ways to quantify complexity. So for this video, it's Kolmogorov complexity and its implementation, but don't hesitate to replace like this blob of Kolmogorov complexity with something else like a Shannon entropy. And you're going to see in the paper that we're going to review, they actually replace this blob with like 3 different complexity measures. All right, perfect. So let's dive into Kolmogorov complexity now. We'll make this part fairly short since I already have a full video walkthrough about all of the theory behind Kolmogorov complexity, which should appear as a card over there. To check it out if you have like 10 minutes, but this will be a condensed version of it. So the definition of Kolmogorov complexity is stated as follows. So the Kolmogorov complexity of an object such as a piece of text is the length of a shortest computer program in a predetermined computer language that produced the object as output. So it is a measure of the computational resources needed to specify the object and is also known as algorithmic complexity. So what does that mean? In a nutshell, the Kolmogorov complexity is simply the size of the shortest program that can produce the desired object. So you see here why it's a theoretical measure, because the programming language is not defined. So this is important because you can't just use it, it's purely theoretical. So if we take a look at an example of how you could theoretically be used, let's say in a purely abstract computer language. So if you have 2 strings, the first string is like A, B, A, B, A, B repeated 16 times, and another string which is like just random stuff like 4, C, 1, G, the thing that is on the screen. The first one will have a smaller Kolmogorov complexity because you could just write this program, which is write AB 16 times. Let's say this was the programming language, which is fairly small. But the second string, it has a higher K complexity because the only obvious way of writing the program will be write for C, I, then the whole string, which is much bigger than the first one. So in practice, however, the K complexity is simply approximated. So we're using compression algorithm in the following way. So the K of an object is smaller or equal to the size of the compressed object plus the source code of the decompressed function. So this is where the relationship between the complexity, the Kolmogorov complexity, and the compression comes into play, because when you're doing a compression, basically what you're doing is finding new ways of saying the same thing in a smaller fashion, and there's a decompressed function over there that is able to take this and reproduce the output. So this is where the connection is. So this is practical because we know that a compression algorithm will have an easier time compressing something with a very low complexity than something that is highly complex. That's basically how they're made. So by taking the size of the whole archive generated by a compression algorithm, we have a good upper bound approximation of the key complexity of an object. So let's briefly touch on a real experiment involving Kolmogorov and DNA to see what it looks like in practice. So the paper we'll be looking at is the following one. It's called Comparison of Complexity Measure for DNA Sequence Analysis. This is exactly what we're trying to go to, and it's by some researcher in Costa Rica. So this paper you can find on my GitHub. I've put it into the repository. So I'll highlight the important bits and share my interpretation of the result. We'll go through the important section mainly to help us implement the thing afterward. So let's take a look at the abstract. So this says, this paper looks into DNA analysis by computing and comparing complexity measure, in addition to providing a review of recent study regarding the measure of DNA complexity. So the other compare Shannon entropy, Kolmogorov complexity, and statistical complexity. And observe the region to the and observe that regions corresponding to gene have constantly different complexity measure than those regions that do not have any gene associated with them. So this provides insight on how to develop new tools for automated DNA analysis. So aka there are other measures of complexity than K that can be used, and the K complexity is approximated using Lempel-Ziv, which we'll see in the next section. So next bit that is important is this one. So Gusev and others evaluate genetic complexity by finding the amount of repetitive sequence. So with a Lempel-Ziv measure, the role of protein coding DNA and regulatory DNA has been understood only recently and provide insight on the belief that organism complexity is related to amount of extra DNA. So here, this bit means that K complexity is a good proxy for an organism complexity from past research. So therefore Lempel-Ziv, which is a good proxy for K complexity, should be a suitable proxy. So the next bit that is important is, for the purpose of this study, the authors choose a short sequence of genomic data taken from the generic Human Genome Project of 384 kbp. Specifically, the data correspond to chromosome Y, some part of it. So this means that their data is all coming from the Human Genome Project. You can browse the Y chromosome on the NCBI website if you want. Good. The next part that is important is this one. A moving window, which they call local profile. Analysis of 250 base pair per computation was performed, and a graph of the variation of the specified complexity measure along the full sequence was produced. So to translate, they used a window of 250 nucleotide to calculate one metric of K complexity, or Shannon entropy or statistical complexity. And then they say the analysis window was moved 50 base pair after each computation in an overlapping manner. So what they mean here is that they took one metric of 250 nucleotide. So they took these and they generated one metric, and then they shifted that by 50 base pair and they repeated to get one other metric. And those are all overlapping by about 50 base pair. So they repeat that analysis over and over until they hit the end of the chromosome. So that thing that they did allowed them to plot all of these complexity metrics across the whole length of the Y chromosome and create a sort of kind of a spatial complexity graph. So part of the graph which has spike is more complex, and with where you have trough, there's less complex. This is what we're trying to produce. So it's not just one metric, so a metric across space, across the space of the chromosome. So that's what we have. The input is a bunch of letter A, T, G, C, and then all mixed up. And the output is a graph of complexity across space. So let's take a closer look at this metric to understand how to use it ourselves. So Lempel-Ziv complexity stems directly from the LZ type of lossless compression algorithm. Two of the popular ones are LZ77 and LZ78. So there's a lot of quite neat YouTube video on this subject. So I'll link them into the comment section so you can take a look about how those compression algorithms actually work. Basically, to get LZ complexity, you just need to take this algorithm and pop off the saving of the compressed information. If you just do that, you're able to record the LZ complexity throughout the reading of the whatever sequence. In practice, this is good, but it's a bit overkill since even with the compression information, we get the good upper bound of the K complexity. So technically, you can literally zip something with LZ77 or 78, and then the size of the archive is basically your complexity measure. So it need to be noted that the LZ complexity work on stream of bits for the compression. So what is imputed need to be a sequence of 0 and 1. So if you remember, we said there was ATGC, but here for this part we need 0 and 1s. We're going to see after that how to handle this transformation. Roughly, LZ77 work as follow, given a stream of bit read from left to right. So first you read the first bits, and then you put in exhaustive memory or not if you already see the pattern. So as you're reading to the bits. And then what you do is you find the maximum component from the exhaustive memory to explain the data that is appearing on the right of where you are. When a streak is broken because you cannot find something in the exhaustive memory, you put the new component that you found inside the exhaustive memory. And then basically the complexity is just the size of this exhaustive memory. A simple Python implementation of LZ complexity will look like this on the screen. So there's a beautiful implementation of it by some guy called Nareen on GitHub. So I linked the thing in the code base that I'm using. So you should definitely check it out. Actually, the documentation on this thing is pretty neat. So it's a fairly short algorithm. And in this particular implementation, the substring generated are actually kept. So you can print them out if you're curious about like what are the bits that are in exhaustive memory. So if you want more details about Lempel-Ziv, feel free to check out the video that Charlotte did that should appear as a card again. I went very deep into the algorithm and the implementation, but that's basically it. There's nothing more than that. You have bits of 0 and 1s, and you're checking for components which you can put in exhaustive memory, and then you can assess the size of this thing. And the size of this is the complexity. Let's check out the data pipeline we'll be generating now with our knowledge of the whole analysis. So with the information that we've gathered from all of these different sources, we can craft a pretty good data pipeline, which we're going to understand clearly the component that compose it. So we'll follow the previously shown conference paper so as to generate something that is similar to their spatial complexity graph, given the input, which is the ATGC sequences of character. So let's break down each component after we've loaded a DNA sequence of raw character into our code. So the main part of our code has to do with calculating the spatial complexity graph. So to do so, we will need to repeat analysis that will calculate multiple K complexity metric across the length of the whole DNA sequence. So the first step in the loop is to create a 250 nucleated window where we are where we're at in the DNA sequence. So then we need to take this 250 nucleated sequence and encode that into bits of zeros and 1, because remember, Lempel-Ziv needs 0 and 1 as input. So we do this simply by converting the characters into their bits representation. So what we get out of this manipulation is a series of zeros and 1 representing the 250 nucleated subsequence. So we then take this sequence of bits and feed that into our code that we've seen earlier to calculate the Lempel-Ziv complexity. What we're going to get out of the Lempel-Ziv complexity applied to this sequence of 0 and 1 is actually one metric that will determine the K complexity for this 250 nucleated sequence. We store that information. So then what we do is simple. We take this window and we shift this by about 50 nucleotide to the right, and we just loop back again. So once we have run through the full length of the sequence, we normalize the K complexity series we've created to see how it differed across the whole DNA sequence. So we kind of normalize it throughout. That's it. The pipeline is pretty simple. So in order to get the data from the raw ATGC sequence into a metric graph, at the end there's all of this manipulation to generate a nice figure, but what you're going to get is an array of K complexity, and this is pretty simple to plot. That's it. There's nothing else really. Let's tie this into some very basic Python code and show how to produce a similar kind of graph we've seen earlier. So once the whole analysis is mapped out, it's pretty simple to construct a working code analysis without too much trouble. There's always optimization that can be done, but I like to start my analysis with an easy to understand state and then optimize afterward for performance. So yeah, this thing is not optimized. Don't forget, you can find the full code in my GitHub, by the way, so you can follow along. All right, perfect. So here we are in the code. So this is regular Jupyter notebook. We're gonna go through the whole thing. So the very first part of this thing is to load the data. So what I did is I took this DNA sequence from this DNA sequence dataset. I didn't use the Y chromosome like they did in the paper. This doesn't really matter. I didn't want to mess around with the Human Genome Project and download all of the data that is enormous. I'm just taking like some small DNA sequence which look like this, and use this as a proxy for a full chromosome. So I've downloaded the data, put it into this nice on my desktop, and then what I'm doing here is just I'm literally just reading the CSV file here, and then you can see it. So the very first step, we read the CSV and we get this. So we have one sequence, another sequence, another sequence, and there's some class associated to it, but like I said, it doesn't really matter because I have no idea what this data set is. What really matters for me here is just like these snippet of character that kind of look like a DNA sequence. So at this point, that's it. We have loaded the data. So that's the first part of our pipeline. So the second part is to set up all of the helper function that we're gonna need in order to use our pipeline. So in the image that I showed you earlier, it's all of the yellow boxes. This is what I've coded over there. Just here, usually when you plan research analysis or some kind of thing like this, you see that when you plan it and when you do it, some of the boxes that you think should be like something are actually blended together, or something that you think were blended together are actually dissociated. It's fine to not follow exactly the plan, but the purpose is just to create a proper mental model before coding anything. If you go straight into the coding, it's going to be a mess. Having a plan, high overview of your analysis usually help when you're dealing with data science project. All right, cool. So let's dive into the helper function one by one. So the very first helper function is this one, is the Lempel-Ziv Complexity metric. So the actual source code for this is over there. I just copied and pasted it, but you can pip install it or something. Do check it out. The source is pretty neat. But that's it. So you accept the binary sequence, and given that binary sequence, it will go through and just calculate the Lempel-Ziv complexity by looking at the component that we have. And you can actually check out what are the component because it's are in the substring over there. And like I said, at the end of it, this is kind of your exhaustive memory, and you're gonna take the cardinality of this, and you're just gonna output it. And this is the length of the substring array here. So the second helper function is string to binary. So I remember the Lempel-Ziv complexity that we saw earlier takes zeros and one, and what we have is literal letters. So what I'm doing here is I get the string sequence, and then I iterate on each of the character of the string sequence, and then I format them so that they are in binary, and I just join them all together. So basically I'm gonna get like a bunch of zeros and ones, and then a bunch of zeros and ones all stitched together. Right, so the third helper function that we have is our generator to generate each of the window that we need, which basically will just slice up the stuff properly and always return us the right window. So here I decided to use yield in the generator, accept a sequence, so a sequence of character in this case, and then a window size 250. This is what the paper was using, and then a jump size, which is like how much we're just gonna go if you have 250 window and then 50 jump size. You're just gonna have overlapping things. Yeah, pretty standard stuff over there, and I'm just yielding it, so I'm using it literally as a generator. I could have used a for loop. Yeah. So my next helper function here is generate LZ sequence. So what I'm doing here is I'm giving the sequence, I'm gonna generate all of the nice windows I need, and then I'm gonna call all of the other function that I need. So I generate the window sequence, so I have this whole generator thing going on. I get my window, I encode the window with the string to binary function we saw before, and then I get that encoded sequence. I've put it into the Lempel-Ziv, and finally I just append this to a LZ sequence kind of array, and I return that. So one of the last helper function I have is normalize LZ sequence. I take a LZ sequence with the window size, and basically I'm going through all of the Lempel-Ziv complexity metric, and I just divide by window size over here. Pretty sure there's a better way to do this than what I did. So the very last helper function I have, I deleted it. So basically here, the last thing I'm doing is just plotting the whole thing. So given a normalized LZ sequence, I'm just going to go through the whole thing and generate this beautiful graph, a bit squished, and yeah, this one's a bit squished, but basically what it is the Lempel-Ziv complexity over all of the 250 windows that we have. So it recreate kind of the paper graph, and you see here I'm doing it at different resolution so that we'll be able to zoom in and out of the thing. All right, perfect. So now that we have all of these helper function, it's time to stitch all of these together. Just a side note here, I really like to work when I'm designing an experiment like this on each of the components first before diving into the mega analysis and then doing the actual thing I need to do, because it helped me out understand like the structure of my analysis, if something that I thought was easy is not, where is the in and out of everything, and also to test each of the components independently. So after that, you're gonna see it stitching everything together is usually like fun because the work is done. You just need to put all of the bits together. Yeah. So the very first thing that we did here is actually we simplify a bit the thing. We've stitched together the data frame. It was this weird thing over there with the different sequences and classes. I've just dropped the classes and got all of the sequence, and what I did is over here I'm just putting that into a list and just joining everything into one mega DNA sequence. So it's very big, but it's kind of like to simulate a Y chromosome. Yeah, at this point I could just have loaded the Y chromosome and done it directly on it, but yeah, so perfect. So that's the disregard Shannon entropy. So here, what we're doing is we're calling generate LZ sequence from the mega DNA sequence, and you see we don't have to put the parameter because that's literally what that's the default variable. So 250 and 50, and then we just get that DNA sequence, that LZ sequence, and we get that length of that number of metric that we've got. It makes sense that is way smaller than the other one because you have a 250 window that you jump by 50. So yeah, if you were to get exactly a similar size or roughly similar, you will have to do like one nucleotide jump. That will be really there will be a high resolution, but will take a long time to generate that thing. And then what we're doing here is just we normalize the sequence. Actually, I could have baked this into the generate LZ sequence, but whatever. And I'm using 250 window size, and I get a normalized LZ sequence over here. Actually, this could have been a parameter that is, yeah, I could have done this. So I don't have to output that because the window size is the same. So when I'm here, then I'm generating multiple graph at multiple resolution. So what I'm doing here with the resolution is kind of simplistic. I'm just getting either the first part, right? So from 0 to 100 LZ sequence, or 0 to 1,000 or 0 to 10,000. Yeah, that's what I'm doing over here. I'm creating tree graph from with 100, the first 100, the first 1,000, the first 10,000, and here I'm getting the whole sequence. And the whole sequence is this. You see, it's very difficult to see. I could have made a prettier graph, but that's what we were playing with. So that's it. With this, we should be able to generate some cute picture of the Lempel-Ziv complexity across the DNA sequence. So by extension, the Kolmogorov complexity. Let's take a look at them. So this is Lempel-Ziv complexity across DNA sequence. This is the first 100. So you see it goes from 0.91 to 0.96 and kind of go up and down. That's pretty much that. And then this one is also, this part over here is this little part over there. This is with the 1,000 Lempel-Ziv complexity point. So you see you have a bit of up and down. You have always oscillation here, and then there's some part that is way less complex than others. And if we take, this is for 1,000 and this is for 10,000. You see here, this is where we're at. This is this one. Even though that this was a bit rough, there's still bigger trough into our whole stitched together DNA sequence. And if you take the full thing, you see that there's bigger trough over there, and then things that are much more complex. So what does that mean? These are technically more random, and these are technically less random. That's it. So the output of the graph actually looks somewhat similar to what's in the paper, where there's many differences in the sense that I feel like normalization that they've done and I have done isn't exactly the same. It's a bit difficult and opaque to know exactly what they did, because they didn't exactly say how they did it. They just say that they were normalized it. But this is what happened when you publish a paper without any code. So you see their stuff goes from actually it's very close to 0.3 all along, and mine is closer to this. I'm pretty sure that they normalize into like bigger window, or maybe the whole Lempel-Ziv. Honestly, I'm not sure how they normalize to get it centered or around here. But yeah, so yeah, like this thing, there's segments which are relatively way less complex than others. In this stitch up sequence, kind of seems to make biological sense. However, we can't know if the dips that we see here are just like significant or not. What we actually need is control, which is pure synthetic gibberish, which is random sequence. So for demonstration purpose, like we're able to do it, but like you will be able to see in the paper, they actually did a statistical significant test with like the actual sequence from the Y chromosome and just a bunch of gibberish that they've set up, and then they will be able to tell if like the dip that we see are significant or not. So yeah, if you're interested, I would argue that you should read the paper in depth, because there's kind of a twist. They've seen that Shannon entropy and the statistical complexity were actually significant and useful to check which DNA sequence were encoding protein or just regulatory DNA. However, Lempel-Ziv was not, and they argue over there that it's because it's a measure of algorithmic complexity, while Shannon entropy and the other statistical complexity is a measure of informational content. So there's a difference. You remember at the beginning I said like Kolmogorov complexity information. The link is not the link between the two is not too clear because it's algorithmic complexity. It's not information content. But yeah, do read the paper. It's pretty good. So that's it for today's video. I hope that it was useful for you. If you found something that I messed up, because for sure I did, or something wasn't clear, don't hesitate to reach out to the comment section. It's always useful for me to get feedback and to improve on them. Also, if you're struggling with a particular topic around the theme I've covered over here in this channel, don't hesitate to shoot me a message or email me. I'll be happy to help out if I have some time. Usually I will not make a whole video, but if like the content is interesting enough for a larger audience, for sure I'm gonna share this with more people. So that's it. Like always, have a great week.